The next system that we want to look at is the vestibular system, or our sense of balance. This system is also going to be located in the inner ear. So if we look at the structures associated with the vestibular system, for orientation we show the cochlea and how the auditory nerve, or the branch of cranial nerve 8, exits. We are going to see, first of all, two structures associated with the vestibular system. The saccule is located on the bottom. The utricle is located on the top. These two structures together are sometimes referred to as the otolith organs. The otolith organs are going to be structures that use calcium carbonate crystals in the fluid to help the cells depolarize. The utricle and saccule are for linear acceleration. Then we will see three semicircular canals. There is an anterior, a posterior, and a horizontal semicircular canal. These three canals are set up in the X, Y, and Z planes of the body. The entire vestibular system is filled with endolymph, and you may remember that the scala media of the cochlea is also filled with endolymph. Notice that the cochlea and the vestibular apparatus are connected to each other. This structure, called the cochlear duct, allows very, very loud noises to cause the endolymph of the scala media to transfer energy into the vestibular apparatus. This is why some people, when they hear a very, very loud sound, will actually experience vertigo. The semicircular canals are going to sense angular acceleration. Coming from the vestibular apparatus, we're going to see the second part of cranial nerve 8, which is the vestibular nerve. The vestibular apparatus also uses hair cells in order to sense fluid movement. So when we look at these hair cells, we're going to see, first of all, a very, very thick, long fiber, which is called a kinocilium. Then, next to that, we're going to see a series of thinner, shorter stereocilia. The way that this system works is that the kinocilium serves as the reference point. If the stereocilia bend toward the kinocilium based on fluid movement, this cell will depolarize. But because of fluid movement, if these stereocilia move away from the kinocilium, we will get a hyperpolarization of the hair cell. From the hair cells of the vestibular system, we are going to see cell bodies located in the vestibular ganglion. The vestibular ganglion is going to have two parts to it. It's going to have a superior section and an inferior section. And what makes these two areas different is where the nerve fibers are coming from. Notice that the superior section is going to have fibers from the saccule, the utricle, the horizontal and superior semicircular canals, whereby the inferior part of the vestibular ganglion is going to have fibers from the saccule and the posterior semicircular canal. Regardless of whether it comes into the superior or inferior, the axons are going to come together. They are going to form cranial nerve 8 and enter into the vestibular nuclei. You'll remember from our discussion of cranial nerves that there were 4 vestibular nuclei: superior, lateral, inferior, and medial. You will not be responsible for the inputs into these different nuclei. But what is important is the outputs. So coming from the lateral vestibular nucleus, we are going to see the formation of the lateral vestibulospinal tract. We will see in just a moment when we look at the connections that this tract innervates the muscles of erect posture, and this is an excitatory tract. We are going to see from the medial vestibular nucleus the formation of the medial vestibulospinal tract. This tract is going to go into the neck muscles, and it's going to be an inhibitory tract. Coming out of the superior and the medial vestibular nuclei, we are going to see the medial longitudinal fasciculus, which is going to be for the vestibulo-ocular reflexes. So what we are going to look at in the next 3 slides are how the vestibular nuclei are going to form various tracts such that we can keep an upright stance. Our first tract then is going to be associated with the vestibulospinal tracts. First, we look at the hair cells. Again, for testing purposes, all that we want to do is identify this as a hair cell. I am not going to hold you responsible for where the hair cells come from. These hair cells have cell bodies in the vestibular ganglion, and they are going to project through cranial nerve 8 to the lateral vestibulonucleus. Coming from the lateral vestibular nucleus, we are going to see the formation of the lateral vestibulospinal tract. The lateral vestibulospinal tract is going to come down. It is going to synapse in the spinal cord, where I will see motor neurons coming out to the muscles of erect posture, and this will be an inhibitory tract. Notice that this tract is an ipsilateral tract, meaning it stays on the same side. Then we can also have hair cells with the cell bodies in the vestibular ganglion going into the medial vestibular nucleus. Coming out, we will have the medial vestibulospinal tract. These fibers will stay on the same side, but also some of the fibers will cross over the midline and go to the opposite side. From here, we will have motor neurons going out to the neck muscles of both sides, and these will be inhibitory neurons, so that the way that we maintain an upright stance is these 2 tracts battle against each other. And by doing that, we maintain an upright position. If we are going to have motor innervation, we need motor system control. So our next tract is going to show how the cerebellum can affect the lateral and the medial vestibulospinal tract. So first of all, we are going to have neurons that are going to come from hair cells of the vestibular apparatus. We are going to have cell bodies in the vestibular ganglion. We are going to have these fibers come through cranial nerve 8. They will loop through the vestibular nuclei and go into the cerebellum. These fibers allow the cerebellum the current status of vestibular activity. Next, coming from a vestibular nucleus, In this case, I'm showing this coming from the lateral vestibular nucleus. I'm going to have nerve fibers that also come into the cerebellum. This will tell the cerebellum the current status of the vestibular nuclei. And then finally, I have neurons coming from the cerebellum going back out to the vestibular nuclei. This is the fiber that is going to have the effect on either the lateral or the medial vestibulospinal tract. All of these fibers pass through the inferior cerebellar peduncle. Finally, we know that in the human, eyes and balance are tied together. So what we are going to see is we are going to have hair cells located in the vestibular apparatus. We are going to see that these hair cells have their cell bodies in the vestibular ganglion. These fibers are going to use cranial nerve 8 to enter into the medial and the superior vestibular nuclei. Coming out of these two nuclei, we are going to see nerve fibers that come together to form the medial longitudinal fasciculus. These nerve fibers are going to go into the cranial nerve nuclei that are associated with eye muscle movement. So from top to bottom, we are going to have the ocular motor nucleus, the trochlear nucleus, and the abducens nucleus. Notice that this is a bilateral innervation. So back when we were studying the brainstem and we identified the medial longitudinal fasciculus, and we always said that it had to do with eyes and balance, now we can see how that is true.